Assalamualaikum warahmatullahi wabarakatuh. Today we will present about a simple method for determination of caffeine content in tea and coffee, and this is our group members.
What is HPLC? HPLC is a type of chromatography that employs a liquid mobile phase and a very finely divided stationary phase. To obtain satisfactory flow rates, the liquid must be pressurized to several hundreds or more pounds per square inch. The objective of this method are to determine the retention time of a standard solution, to identify the caffeine peak in a soft drink sample, and to determine the amount of caffeine in soft drink sample using the response factor method. We will go to the methodology. So this picture shows the whole system of HPLC system, and next I will explain detail about its function and how it functions in this experiment. Okay, so next we go to the function of each component in the HPLC system. So the first is solvent reservoir, which is it holds the solvent mixture used to carry the sample through the system. And the second is pump, pressurizes the solvent mixture and controls the flow rate, ensuring a steady and consistent delivery to the column. The third is sample injection, introduce the sample into the flowing solvent stream. And HPLC column, the heart of the system, where the separation of components occurs. The column contains a stationary phase, for example silica gel, that interacts with the sample components, causing them to separate based on their affinity for the stationary phase. And detector is to detect the separated components as they elute from the column. Different types of detectors are available, such as UV-Vis, fluorescent, or an electrochemical detector. And the second last is WASE, which is collects the eluting solvent after the detector. And lastly is PC, which is it ensures accurate, reproducible, and efficient data management, making it an essential component of modern HPLC system. So in this experiment, we use HPLC to determine the caffeine content in tea and coffee samples. The process starts with the solvent reservoir, which supplies the mobile phase a mixture of water and methanol, 60/40. This mobile phase is pumped into the system using a constant flow rate of 1 ml/min. The prepared samples, filtered through a 0.2 micrometer microfilter, are injected into the system through the sample injector. And then these samples pass through the HPLC column, a Zorbax Eclipse plus C18 column, for example, which separates the caffeine molecules based on their polarity. This separated caffeine is detected by a UV detector set as 275 nanometer, for example, and the resulting chromatogram is sent to the PC for analysis. And then, using a calibration curve of caffeine standards, we calculated the caffeine content in the samples which met regulatory standards. This method is precise, accurate, and validated for routine caffeine analysis. Okay, so in this experiment, we use
From the result, you can see there's two figures of chromatogram. From figure 1, the figure shows that the height of peak of caffeine at 2 ppm. The peak shows only peak of caffeine in standard caffeine. As for figure 2, the figure shows that the height of peak caffeine in tea sample. The chromatogram show that there's two of peak represent caffeine and tea. Larger peak correspond to higher concentration, while smaller peak indicate lower concentration. Next, the graph show that the calibration curve of the concentration of the caffeine standard where concentration of the analyte are plotted against respective peak areas. From the result, it showed that the standard solution was repeated 6 times. This makes the retention time more accurate in further analysis. It is observed that the sample peak retention time to standard caffeine is around 2.6 minutes. To determine the amount of caffeine, HPLC are used for further analysis. HPLC stands for High Performance Liquid Chromatography. It is a strong analytical method used to separate, identify, and quantify the components in a mixture. The chromatography shows both peaks are relatively symmetrical, indicating strong separation with little interference from other substances. In the context of chromatography, retention time is an important quantity that specifies how long it takes for a certain molecule to transit down the chromatography column and be identified by the detector. From the result, it showed that retention time is 2.6 times, indicate that the substance in the sample is relatively nonpolar. This is because nonpolar molecules interact less with the polar stationary phase and dissolve more quickly. A calibration curve is the graphical representation of the relationship between the measured signal for the peak area from the HPLC chromatogram and the known concentration of a substance. This calibration curve is important for quantitative analysis since it allows us to calculate the concentration of an unknown material using its peak area. The R-squared value of 0.9928 indicates a strong linear relationship between concentration and peak area, allowing for accurate measurement.
To conclude, all the objectives have been obtained. The retention time of the standard solution is 2.6 minutes. Next, the caffeine peak in a soft drink sample is identified in Figure 2. And lastly, the amount of caffeine in a soft drink sample using the response factor method is determined, where the average value is 98.27%. That's all from us. Thank you.